Hi, my name is Enrique Velazquez Villarreal. I'm working in Keck School of Medicine, USC, Department of Translational Genomics, working with Dr. John Carpenter and Dr. David Craig. And basically we have a collaboration with 10x Genomics. This kind of information is basically leveraging new methods in single-cell copy number analysis and clonotype detection just to characterize hiding subclones within the standard cell lines. At this point, just a little bit about background. So single-cell sequencing has become a very powerful tool, but largely studies have focusing on single-cell RNA-seq. What we are using here is a new technology, Chromium TM from TX. This technology is processing thousands of single cells. And at some point, it's very interesting that this technology is able to call clonotypes down from 1% from any 1,000 input cells. And this is interesting because from these 10 cells, we can check for 100 KB events. And that's very interesting to understand cancer heterogeneity. So very important here, our methods. Our pilot study processed 1,446 single cells using standard cell lines, Clono 829. Through the system, we generate barcode of each cell. At some point, using bioinformatic analysis, we can generate information from each of the cells. Interesting, we use this barcode analysis in order to detect copy number variation. After we detect this copy number variation, we generate this kind of graph. In this case, it's a dendrogram with a heat map. Interesting in this heat map, each line represents one cell. If you see on your y-axis have the number of cells from 0 to 1,446 single cells. So it's supposed that in bulk sequences, you should have the same color in any chromosome, means the same copy number. Here, it's something different. Since we are going to single cell level, we can check each of the cells and the heterogeneity of the entire sample. This means from these cells, we did unsupervised clustering, means that create 6 different groups. Then we check the difference between groups. If you see, group A is having 2 copy numbers, but then the other groups in 1P level have 1 extra copy, 3 copy numbers. So different happen in at Q level with all the cells, or most of the cells, have 2 extra copies. This heterogeneity is very important since we can check for events that lead different behaviors in copy number variation. For example, chromosome 8 here, it's led by a big chunk of 2 extra copies at P at Q level. And it's basically describing our group C. So by going beyond this graph, we can create kind of a history of different events that happen in copy number variation. So for example, here in group A, what we are having is diploid at P level, but then 1 extra copy in the other groups. Interesting, by checking this heterogeneity, is that group A have at Q level 2 extra copies, but then group B has 1 more than A, so 3 extra copies of Q, and then F have just 1 extra copy. So interestingly, this is something that we cannot detect using bulk sequencing. So at the end of the day, we have right now showing the ability to better resolve absolute copy number, and whereas previous analysis approaches using bulk sequencing have lagged due to the reliance of indirect inferences. So we are more right now focusing on direct results. So overall, all results suggest a single cell copy number provides a unique and important insight into larger scale events like a P or Q deletions or insertions, or amplifications. So providing unique and distinct insight into cancer heterogeneity. If you have more questions, please go to 10X website. And we also are very happy to collaborate with you at USC Department of Translational Genomics. Thank you so much.